All of them are very health impacts, carcinogenic, neurotoxic, and we've been talking a lot about them after the writing of Rachel Carson on Silent Spring. So this I will skip because you all know about it. I'll just come to the point how anthropogenic activities as well as the associated impact factors of environmental, physical, chemical, biological contamination these POPs get spread. So there are in fact scientific basis like Maitri research base and this is a Bharati research base, then another Progress research base, and then this is a Chinese base, and there are many tourist activities which causes these contamination levels high in the and this is the orographic effect besides the anthropogenic and scientific activities, because I hope most of you are aware about the grasshopper effect, how the air rises from the warmer climate and that condenses into the colder climate and it brings the POPs. This is about it. So according to our research in this area, basically northeast area is most contaminated with POPs, and this is west and also southeast area. So these are the larger pictures, the points where we have found more persistent organic pollutants. So these are there in water bodies. You can see the levels are as high as 2300 picogram per liter. These are polycyclic aromatic hydrocarbons, PCBs, hexachlorohexane, PCBs, and PHs and PCBs. Next, in air sample, you can see 20 to 43 picogram and Antarctic Peninsula. I have made them red in color, which are high levels, but these are also levels which are beyond permissible limits. And these are all picogram levels. So again, I am talking about PBDs, PCBs. Now in Antarctic sediments, and soil accumulates more of these persistent organic pollutants. So you can see the level up to 1100, 2100, 1077, 2000, and these are from various areas of, I have already shown you the map where these have been found. Now it has been also, besides sediment, soil samples also have been analyzed. Again the levels 46,000, over 46,000 nanogram per gram. So this is very high. Picogram, this is by in nanogram, you can see how high the concentration is. It is in McMurdo station. Similarly in Victoria land, it is 112 to 561 picogram. Now in our sea ice and snow, you can see 110 to 580, 130 to 340 picogram. This is an Antarctic mosses. This is now the biotic component is coming. Soil, water, snow, I have taken air. This is in the mosses around 1200 to, they basically it gets biomagnified, so it is more in the biotic component, 1620 to 580 picogram per dry weight. Similarly, in lichens, 353 to, and this is also 207, 632 picograms. So you can see these PCBs, which has nothing to do in the pristine environment of Antarctic, but they have found their way in the pristine environment. Again in Ross Sea in Terra Nova Bay, 1.6 nanogram in Antarctic krill. In penguin eggs also, and penguin around 153 nanogram per gram. And we have also analyzed, this was the literature review which we have done in POP sample during the 35th and 34th and 35th ESA. We are included in the environmental monitoring program. So we took samples from the Brookness and the Brookness Peninsula. We have the Bharati station here. So these are the Brookness and Brookness. And we took sample from, there are many lakes, from 20 lakes around. There are some of the beautiful pictures. And this is the procedure which we followed for the extraction cleanup. Methylene chloride and then pulled in and then extracted with hexane. So our detection limits were 0.01 micrograms. You can see how much it is, 50 nanogram, 40 nanogram, 20 nanogram. And it comes to picogram, it will be around 40,000. So the levels are quite high. The one which we could not detect because we have limitations with our detection limit. In other countries, they have detection limit up to picogram, but we have a limitation of into microgram, 0.01 microgram. So when you convert into picogram, heptachlor, alachlor, these both are herbicides and they are being sprayed aerially in the fields. So basically we have expertise in the pesticides analysis. So monocrotophos, 4-8, we have taken all the variety of pesticides and a very complicated analysis procedure all these pesticides have. But the categories are main organochlorines and organophosphorates. But we have also taken the herbicides, alachlor, isoproton and these. Even naloxone, this is a pesticide, you can see. So this comes up with 90,000 picogram. Malathion, chlorpyrifos, 4-8 sulfoxide, these are the metabolites of 4-8, even the metabolites like DT is metabolite. Even their metabolites are being found in the Antarctic lake water samples. Then we also analyze polycyclic aromatic hydrocarbons. So you can see even naphthalene, though it is not much carcinogenic, but still it finds its way in the lake water samples. Fluoranthrene. Then these are all not detectable. So conclusion is that they have been found to be highly varied between different regions of Antarctica, and in fact the food supplies which are reaching Antarctica may carry these pesticide residues, and occurrence of these POPs may cause mutagenicity, genotoxicity, reproductive disorders, disruption of immune system and interferes with developing of the young. I am talking about the fauna in the Antarctica. So, in fact, we should check, since we have analyzed pesticides, there should be proper enforcement for pesticide regulations and effective monitoring by apex agencies for banned pesticides, then proper training for the use of pesticides, and also dosages according to the pest and crop. Basically, there is a method called integrated pest management techniques which are available for each and every crop at the IPM center and CIPM center, where cultural, biological and mechanical control methods are given for every crop and every pest. So those should be followed. Next. Then we should opt for clean energy. Basically, even the thermal power stations, they emit lot of heavy metals as well as PCBs, polychlorobiphenides. So we should switch to the developed countries are coming forward to help underdeveloped countries or developing nations with clean energy like nuclear power and solar power. But nuclear power also again has its own limitations. And that should curb emission and transport of these xenobiotics in atmosphere and thereby in Antarctic ecosystem. And use of generator, vehicle, helicopters, flight, ships and other modes of transport which use fossil fuel also contribute to these POPs in this region. Then personal care products, physical contaminants, construction material also contribute. Then we need to cut down the usage of plastic on Earth because phthalates phthalate residues we have not done in this study. But in later studies will include the phthalates because this is becoming one of the main contaminants. And a comprehensive collaborative doctoral research effort focusing on above factors are urgently required to identify the gap areas of emphasizing the recent development in the POPs research. So, and biomagnification also plays a role in the ecosystem. So, with this view, we opened Antarctic Center for Research and Studies, and the vision was to do the cutting-edge research in the field of environmental contaminants and anthropogenic activities. But later we have also, this is the mission of the PHAs, PCBs, pesticides. I have already spoken that we have, but later we have also extended to the biotech applications such as nutraceuticals, enzyme, antibiotics, surfactants, industrially important products, focus on R&D. We have already grabbed a project on that, on biofuel generation also. I will show you later. So these are our participation in 34th and 35th. This is Anuj is there sitting there. My team is sitting there at the back, who have participated in various expeditions. And then these are the pictures. I'll just skip forward. Yeah, 2 minutes. I'll finish up. Then this is the polar ecotoxicology. I am an ecotoxicologist by specialization. So I am bringing up this field of polar ecotoxicology, which was proposed at the STOX conference organized at MIT. Then we have been participating in Ozone Day celebration and all we are doing. So these are the national, international conferences we have done. We got a project from CSIR on speaking of environmental tolerant poisonous strains from recent Indian station Bharati Antarctic for bioremediation and hydrogen production. And this is another we are participating in NCR in Southern Ocean Expedition. We have one publication in press, and we are getting lot of in the future plans with the Indian expedition, and we are also planning to come up in Arctic also, and we are getting lot of support with MOES. These are the completed projects. And this we have got MOU with University of Saskatchewan. It has an atmosphere like Antarctica, -60 in Canada. So one of my research scholars is studying there, and we are planning studies on ice core. Lot of conferences are organized. You can see Dr. Rasik here on Earth Day, and I'll just finish up by showing. So we have full-fledged laboratories here for research. We have digitalized stations, and this is my team. Thank you so much. I wrote poem on Earth, Sky. I am a poet, though my title is not poetic. So this is about it, and this is also mandates and vision. Thank you so much. Say a word to bring work for the new birth is our punchline. Thank you so much.